So now we move towards facilitated diffusion, which is a type of diffusion in which the carrier proteins are involved. What happens in such a situation is that molecule that is to be transported, it attaches to a binding point on the carrier protein, and the carrier protein undergoes a transformational change and allows the molecule to move either to the interior or exterior depending upon where it needs to be moved. For example, glucose and amino acids, they are transported via such carrier proteins, and the molecules that transport the glucose, the carrier proteins that transport the glucose, they are known as glucose transporter proteins, GLUT proteins, and they are 14 in number. They allow the movement of galactose and fructose. Main one is GLUT4, which is insulin dependent, and whenever insulin attaches to such a GLUT, it allows such a glucose transporter protein. Whenever such a protein is activated by insulin, it increases the movement of glucose and fructose to the interior of the cell by 10 to 20 folds. Now, there are three factors which affect the net rate of diffusion. One is the pressure, second one is the concentration difference, and other is the Nernst potential. Now, the concentration difference, we always know that diffusion is a movement from an area of higher concentration to an area of lower concentration. And in the net case, we can define it as net diffusion is directly proportional to concentration on the outside minus concentration on the inside. Other thing that they have discussed over here is rate of movement to the interior depends on the concentration on the exterior. Rate of movement to the exterior or outside depends on the concentration of the molecule to the inside. Whenever more molecules will be on the outside, they will tend to move inside. Such is the case with sodium. Whenever more molecules will be on the inside, they will tend to move outside. Such is the case with potassium. So now, what is Nernst potential? As we know that across a membrane, we have the diffusion which takes place by the movement of the ions from an area of higher concentration to an area of lower concentration. This is due to their number. But sometimes the diffusion can also be interrupted by the charges of the ions, and in such a situation, we get charge imbalance. But whenever the charge and the number of the molecules, they are balanced with each other, we get a type of equilibrium, and that equilibrium between the number of particles and the charges on the particles is known as the Nernst potential. It is such an equilibrium in which neither the number of the charges are balanced on either side nor the number of molecules are balanced, but there is a type of equilibrium achieved and there is no net movement, and the potential that is produced at this place is known as the Nernst potential, and we can calculate it by EMF, which is the electromotive force, which comes in millivolts, as plus minus 61 log of concentration on one side to the concentration on the other side. Third difference, or third thing that affects is the pressure difference across the membrane. Always remember that the molecules will tend to move from an area of higher pressure to an area of lower pressure, because pressure that is being exerted, it is causing the molecules to be expelled from an area where the pressure is more to an area where they can freely move, that is low pressured area. Now we have the process of osmosis, which is the movement of the water molecule from an area of lesser solute concentration to more solute concentration across a semi-permeable membrane. And what is osmotic pressure? Osmotic pressure is the amount of the pressure that is required to actually stop the process of osmosis, and such a pressure difference is created by the non-diffusible solute ions. And as we know from our previous FSC knowledge that osmosis is a colligative property, it means that it depends upon the number of the particles and not the mass of the particles. And to calculate the kinetic energy, we get this formula. Kinetic energy is half mv squared. That is the same formula. Now we will discuss two principles. One is osmolality and other is osmolarity. So osmolality is number of the solute particles per kg of the solvent, whereas osmolarity is the number of the solute particles per liter of the solution. Now we will move towards a calculation. As we know, osmolality is solute per kg of the solvent, and in our normal body fluids, we have 300 million osmoles per kilogram of the water. Water is the solvent, and solute is having 300 milliosmolar concentration. So, what happens now is that 1 osmole exerts a pressure of 19,300 millimeters of mercury, and 1 milliosmole exerts a pressure of 19.3 millimeters of mercury. So now when we multiply 300 with 19.3, we get a total of 5790 millimeters of mercury. But to our surprise, the normal number that we get is 5500 millimeters of mercury. Why is that so? Why is there a difference of 0.93? This is 0.93 of this number. 5500 is 0.93 of 5790. So our actual value is 0.93 of the calculated value. Why is this so? Because as we know that osmolality is a colligative property. It depends upon the number of particles, not on their size, not on their charge. So in normal cells we have sodium, we have chloride, and we consider them as separate entities. But in our body, the sodium and chloride, they sometimes interact with each other, they sometimes do not interact with each other, and they remain as separate ions. When they interact with each other, they are considered as a single entity, but when they are separate, they are considered as two different substances. So when we calculated it, we considered more number of the particles as were actually present. Hence, we got a larger number than is actually in our body. So this is the normal calculated value, which is 0. So normal value in our body is 0.93 times of the calculated value. In the next lecture, we are going to discuss active transport. Stay tuned.